So, guys, now let's move towards the final part of the metabolism of macromolecules. And now you know that what we are going to talk about? Yes, you are right. It's about the metabolism of nucleotides or metabolism of nucleic acids. Nucleotides are, I don't think that you have heard more about any other compound more than you have heard about a compound which is known as the code of our body. Whatever we perform, whatever we are doing is coded into that. Its information is coded into that compound. And which is that compound? Yes, you are right. It's DNA. And what DNA is made up of? Nucleotides. So now you know how important is to understand the metabolism of nucleotides, how important this process itself is for our body. But you will also see that nucleotides is not all about making the information material like DNA or like RNA. Of course, that's a huge, one of the most important feature of these molecules. But also they play a very, very important role in maintaining the energy dynamics of our body, because they are giving rise to very important energy carrying molecules. They are also, these nucleotides are also very, very important in the formation of some intermediary compounds. So it's not always about the DNA or RNA when we are talking about nucleotides. There is something more to this story. And what is that? Let's see in some detail. So the nucleotides are the fundamental building blocks of our information molecules like DNA and RNA. Without this, of course, they are the building blocks, so they are indispensable for these information molecules. So all the cellular function depend upon these building blocks, because of course, if they are not there, there is no DNA, there is no information molecules, there are no information molecules, the cellular processes cannot take place. They are also very essential for cell division, because cell division depends upon the duplication or replication of the DNA, which again won't be there if there is problem in the metabolism or in the synthesis of these nucleotides. They also, as I have told you, they have role which is beyond their role in the acting as information molecules. For instance, they are also involved in the synthesis of complex carbohydrates, in the synthesis of conjugated proteins or lipids. For instance, the UDP and CDP, they combine with the lipids and carbohydrates like glucose, so to really synthesize complex lipids or the carbohydrates. They provide the trigger for the formation of these molecules, as we have already seen in the biosynthesis of the complex carbohydrates or lipids. In addition, they are also very, very vital and important component of coenzymes like coenzyme A, FADH, NADH, NADPH. You know, these enzymes are involved in all sorts of biochemical reactions, not only for the energy reactions which involve energy, but they are also involved in many other biochemical reactions. There are, you already know, you have been studying it from your matriculation classes, like there are 4 types of nucleotides, and they are in turn divided into 2 types, like purines and pyrimidines. Purines they have 2 circle rings, while pyrimidine has 1. And then DNA and RNA all are almost the same. The only difference in the nucleotides in them is that DNA have adenine and guanine and thymine and cytosine, while RNA have one different pyrimidine, that is uracil instead of thymine. Otherwise, they are very similar. And of course, we will see that as far as the ribose moiety, sugar moiety of a nucleotide is concerned, here we are only talking about the nitrogenous basis of the nucleotides. Of course, then when we talk about the ribose sugar, that's also different between RNA and DNA, which we will see later. Then there is also regulatory role of nucleotides. If there is more the, for instance, the ATP, GTP, AMP, they act as feedback regulators. Depending upon the concentration of these compounds, these nucleotides, a reaction can move in the forward or reverse direction, or can be even inhibited. So, and also they can be, these nucleotides can be produced from the very start, from the very scratch, or they can be salvaged from already present nucleotides. New nucleotides can be formed. They are usually broken down. They are not used as a source of energy. Of course, the nucleotides, they are involved in the synthesis of certain coenzymes, which are involved in the synthesis of, in the formation of energy or energy compounds, but they are directly, their degradation is not used to get energy, as for instance we have seen in glucose or lipids, where the degradation of those molecules is used principally for getting the energy. But this is not the case with nucleotides.